oxygen and glucose. Oxygen and glucose is supplied with the help of these capillaries, which are attached to these myofibrils, which are attached to each cell. So they don't go inside the cell, but they are pretty much attached to each myofibril. And this myofibril can be fairly long type of a microfibril also. It's a compound type of a cell, single cell, but it is compound type that it's made of several different nuclei. Okay, so here's the information again. Myofibril is that thread-like long rod-like structure which is inserted inside each myofibril, each muscle fiber. And myofilaments are those filaments which cover myofibrils. These make the myofibrils. Sarcoplasm is the cytoplasm of the muscle fiber, which is, instead of calling it cytoplasm, it is called as sarcoplasm. Then sarcolemma is the cell membrane. And then T-tubules are transverse tubules which are present inside in a transverse fashion. So they actually are, if this is a myofibril, they are attached around it as a transverse, as a kind of a cross-sectional tubes. Then sarcoplasmic reticulum is endoplasmic reticulum inside the muscle fibers, and their type is of smooth endoplasmic reticulum. Then this is the one that is used for the storage and release of calcium. Calcium is stored here, and it is released from here at the time when muscles contract. The two important proteins are the top two, actin and myosin filaments. So actin filaments are thinner, and myosin filaments are much thicker. And these myosin proteins are in the shape of a club, golf club. So because they have that end which is used for hitting the ball, and these proteins, actin proteins, are straight. They are made of two strands of pearls which are wrapped around each other, or two strings of a rope. Then other proteins, troponin and tropomyosin, are two other proteins which are part of this myofibril also. Troponin proteins are the sites where calcium attaches. So we'll keep on seeing actually the role of calcium. Calcium is very significant when it comes for muscle contraction. So calcium has many roles, and this is actually one of the roles that calcium plays in muscle contraction. So calcium basically binds to troponin protein, and then tropomyosin protein is the protein which is a kind of also a filament. It's also a long protein, just like actin protein also. So it binds to the actin protein. So it is attachment site on actin. So it binds with actin, and it provides a place for binding with myosin. All right. So now this is actually again a single myofibril. So there could be several different myofibrils running through a single muscle fiber, running through a single muscle cell. So again, those two are the two strings of actin protein, and this is one thicker string of myosin or myosin myofilament. Both of those are located inside, and inside they are divided into those small units which are called units of contraction. Those are called sarcomeres. So each myofibril is divided into these small sections which are based on those proteins, those two proteins. In this case, actin protein is shown in purple color, and green color is myosin protein. So from here to here is a single sarcomere. There's another sarcomere here, another sarcomere. So sarcomere is the basic unit of contraction. So this unit is the one that moves the muscles. Since there are several of those units, and these are microscopic, they have to be seen under a microscope in order to see their detail. They are very small, and size-wise they are so small that they kind of attach to each other, and multiple. Whenever a muscle moves, all of them actually move at the same time. all of the muscles which are part of this myofibril, they contract or relax at the same time because they are all attached in tandem with each other. They are actually joined, they are fused with each other, so they cannot really separate themselves from each other. Different muscles can move differently, but sarcomeres, which are present in a single myofibril, have to move collectively. So whenever this moves, it moves in unison, together, all of them at the same time. So now this is the kind of deepest that you can go inside the muscle. Well, we can actually go one more step further after that as well. When we talk about just a single sarcomere, we can actually see how a single sarcomere is organized and how it moves to move the entire muscle. So this is what it shows more detail here. So what we saw in this slide here previously, so this part these two parts are the Z discs. These are proteins, and then within these Z discs there are different zones that are labeled here as I and A. So the Z discs are proteins which are kind of vertical proteins, which are transverse proteins that are present inside each myofibril. And those proteins are attached with these proteins. Now these are again, the two rings or two strands of actin protein. So they are attached like that, away from each other. But they are anchored with these vertical proteins which are called Z discs. So actin filaments, which are kind of attached to the Z disc directly, myosin proteins are attached with this middle line called M line. So this is the kind of middle line where two myosin proteins, which are in the form of bundles, thick bundles, they are attached tail to tail. They are attached away from each other. So one that starts here goes in this direction, the other one goes in the opposite direction. So they are attached opposite to each other on their tails. But these ones are attached and anchored here on the Z disc. This protein, which is called titin protein, basically holds the whole structure of the Z discs with each other because it is attached here and then it is attached to this next Z disc on the other side. So they are attached, and it defines the size of the sarcomere, because Z disc to Z disc is the size of the sarcomere. This is a single unit of contraction, and this is what's inside that small sarcomere inside. Other proteins that I mentioned in the previous slide, tropomyosin and troponin proteins, are these ones that are attached to the actin protein, and these are the attachment sites for calcium, which is we'll see its role later on. So actin or thin filament basically comprises of these three proteins, which is the main part of that is actin protein, and troponin and tropomyosin are the two proteins that are attached and are sites for attaching to calcium. All right, so coming back to, so this is the myosin filament, which is also called as thick filament because of the size. It is made of several different myosin molecules that end with this kind of double heads at the end, club-like structure. So rod is this long filamentous protein which ends with these two heads at the end. So in the middle part of this whole unit is where myosin protein is. So myosin protein is actually a free protein. It is not attached to the Z disc directly. It is attached to this M line, which is a part of the sarcomere, but it is not attached to either side of this Z disc. It is thick, and the way it actually interacts with this actin protein is with the head of its two heads. And these two heads help to contract the muscle. So when there is ATP energy available, the conformation of that head changes in such a way that actually it kind of pulls it, because it is attached like that. So when energy is available, it kind of pulls it like that, and when it pulls it, it brings these two actin proteins towards the M line. So because this is free, these two are attached. So when it rubs against them, by making those cross bridges, when it attaches to the actin protein, that is called a cross bridge. So when it attaches to them, it pulls them like that, and that pulling basically brings this whole unit closer. So then it results into contractivity. So it contracts them towards the middle. The whole unit becomes a little bit shorter. Each sarcomere becomes a little bit shorter, then it relaxes after that, and then it contracts again. So when I'm moving my hand, this is what's happening in those muscles. When I'm moving my arm, this is what's happening. The sarcomeres are shrinking. When I contract it like that, that's what really getting something done. Some work is being done because I'm contracting my arm. So at that time, the sarcomere just shrinks. And when I extend it, then it is relaxing, and they'll go back to their original position. If this is their original relaxed position, the contraction or the contracting position will be when it will be a little bit shorter. And this movement actually creates those striations. Those stripes that we see under microscope are because of this. This unit here should be, it should look darker under microscope because there are 3 proteins here. There are 3 layers of proteins in the middle. There's just 1 layer of protein here, and these 2 are only 2 layers of proteins, which are very thin protein. So you should definitely see some density difference when you stain them with dyes or when you see them under microscope. So when it moves, then you will see actually the whole area becomes thicker in the middle. When it is contracted, there will be 3 layers even towards the middle of this M line also. So it will become much darker in the middle. This area pretty much stays open and does not become stained with the dyes because there is no overlap with the myosin protein here in this area or this area, and they stay pretty lightly colored. That's why the striations are seen under microscope, because of the movement of these muscles. When they move, they become much thicker in the middle. When they relax, that overlap or that kind of a contraction of the protein is completely gone, so they stay a little different. Okay, here are the names again. The sarcomere is the entire unit, which is called the contractile unit. So that's the functional unit of the muscles that contain actin, myosin proteins plus other proteins. We've talked about 2 proteins. The titin protein, which was the 5th protein. So actin, myosin. What are the other 2 names? Troponin and tropomycin. Tropomycin are 4. And then the 5th one was titin. Titin is a long protein. Actually, that's one of the longest known proteins. Titin is a very long fibrous protein, but it is one of the longest known proteins. Here again, those actin proteins, myosin proteins, troponin, which is not shown here, and tropomycin are, and the 5th one is the titin protein. Z disc is a protein. Those are made of proteins, which are these, the vertical ones. Okay, what's in the Z disc? Z disc are protein fibers. What is in the H zone, and I band, and A band, and M line? So zone, band, and line. Okay, disc, zone, band, and line. Z discs are proteins, which are these proteins on which these actin proteins are attached. And then H zone is the center of the sarcomere. So it is not made of protein. It's just a zone. It is just an area to show how they are different. The I band is the band which shows just only the actin proteins, and I'll show you that there. A band is where actin and myosins overlap. M line is where myosins are attached. So when they're attached in the middle. So here, this is Z disc. Again the proteins, and that is the size of this sarcomere. And right in the middle is the A band. A band is where these proteins are overlapping. When myosin and actin proteins overlap, so this is what we see, they become fairly thick type of a band. And then M line is just the line on which the myosin proteins are attached, and H zone is the zone in the middle of this sarcomere when muscle is relaxed. When muscle is relaxed, there is only one type of protein that you see overlap present, which is myosin protein. There is no overlap with actin protein. So that's why this is called that's why it's called an H zone. So it is actually a zone which is occupied or stained with a single protein in the middle. So these striations here, you can actually see those here as well. Let's see, Z band. Z discs are here. So these are the Z discs, that you see with the lighter color, with the darker protein. The line is the Z discs, Z proteins or proteins in the middle that show the darker line. This area of the lighter color is this area here. This area which is stained only with actin protein. So it is not very dark in color at that time. So that's why it looks kind of a little bit lighter. Then this area here, this small area, which is the A band part, which is this part here. So this is the part where these two proteins are overlapping, so it is much darker. And then this area in the middle is the H zone. So that zone is lightly pink color. So this kind of darker pink is this area here, and the lightly color is this thicker filament of myosin in the middle. And this area is called the I band. I band is basically two zones around the Z disc, a lighter one on this side and lighter one on the other side, are just actin proteins. Anybody who's not clear about that? Okay, 1%. Other than 1%. So the structure, I guess you'll have to spend, I can keep on explaining that and we can repeat that several times, but you'll have to kind of draw it out. Just write it down, draw the image yourself, and then label those things by yourself. Then I think it will get a lot more clear to you than somebody keep on explaining that to you. It will be a lot more useful for you to start visualizing it. These are the structures we saw in the video also. Those are the two proteins, or two arrangements on which the proteins are attached in a certain way. And the reason for that is because the contractivity is needed. And that contractivity is done with the help of all those units that are attached in this single chain of sarcomeres. When they contract, the whole muscle contracts. This muscle, this biceps here, is just single muscle. But within that single muscle, there are so many of those myofibrils that are attached to each other, and they all contract at the same time. When you pull your arm, they are all contracting, and they're contracting because of this single unit. So it all starts with a single unit, and then there are more detail after that. Again, this is the kind of sequence that you see. This is the innermost part of the muscle, and then that muscle is made of these myofibrils. Myofibrils are made of myofibrils are inside the muscle fiber, which is the cell, and these are located inside that each cell, and there could be multiple cells that are located. One thing that you probably got it, I'm not sure if you got it from the video or not, when we are born, at that time, the number of muscle cells that we are born with pretty much stay the same number in our body. So as we grow older, the length of those muscle cells continue to increase. And it really does not change a whole lot with our age. The cells continue to grow. The same cells, they continue to grow and become much longer cells. Should we move on, or any questions?
I was bringing up on my blackboard. It's one that makes it real simple that she showed us in biology. That it's so simple that you'll get it. But I don't know how to get it on there. But
I have an animation too. If animations help, if you just Google it, a lot of people have actually made those videos. A lot of publishers have made a lot of videos to explain it, because this is one topic that is always discussed. That's always the one that people have a lot of difficulty with. So muscles, okay, since muscle cells, muscle fibers are cells, so they are just like any other cells, but a little bit different in terms of their activity and in terms of their content. Otherwise, they are just cells. So these cells are excitable cells. They can be stimulated. Stimulation can come from electric current, or it can come from stimulus or chemical stimulus coming from the brain. So their relationship is explained with the help of some channels. Since these muscle cells are related to kind of current that they contain in, all cells of our body have generally two. How should I explain that? Let's see. So the cells have several things on those cells. They have different channels, many different types of channels that provide them some control over what goes inside and outside of the cell. And then they also have differences in terms of what is present in the inside of the cell versus on the outside of the cells. For example, we have more negative charges inside the cell, and there is more positive charge on the outside of the cell. And this charge difference is responsible for a lot of movement, for a lot of the difference in internal environment versus external environment. So cells are generally negatively charged on the inside. So this difference in charge, in the charge or net charge inside and outside of the cell, because of the presence of different types of ions, we actually have some other proteins which are negatively charged. We have DNA inside the cells also which are negatively charged. So there's more negative charge in the inside of the cell than the outside of the cell. And this charge is maintained with the help of these channels, which are kind of holes present in the walls or proteins in the walls of our cells, and there are two main types of those channels. Those channels are proteins that help to move things across the membranes. And there are two types. One is called gated, and another is called leaky channels. Leaky channels stay open. They are called leaky because they always leak. They always continue to have flow through them. They are not controlled. Gated ones are gated. So they open and they close. That's how they control different things that move across the cells. When things are needed to move, then they open. Things can move through those channels. When things are not needed to be moved, then they actually are kept closed, like any gated colonies or any gated neighborhoods. So this is actually from page 47. So something long time ago we briefly talked about. So these are those channels, two types of channels. There are potassium leak channels, and then there are gated sodium channels. So these are the ones that are a leaky channel. They are open all the time. So because calcium is so important, so they have their own channels that can continue to stay open. And then the other channels are the ones which are called gated ones. Gated channels can stay closed, or they can open